In the Antioquia region of Colombia, an extended family network represents the world's largest known familial Alzheimer's disease pedigree. The genetic driver for this specific population is a single mutation in the presenilin-1 gene, designated as the PSEN1E280A, or PISA mutation. For carriers of this gene, the clinical reality is rigid. It triggers an aggressive, early-onset form of autosomal dominant Alzheimer's disease. The timeline is highly predictable across the pedigree. Carriers develop severe cognitive impairment in their mid-40s and progress to complete clinical dementia by the age of 50. For decades, the medical field operated on the linear logic of the amyloid cascade hypothesis. The extreme overproduction of amyloid beta, driven by the PISA mutation, was viewed as an absolute guarantee of rapid neurodegeneration and cognitive collapse. That guarantee failed in one specific heterozygous carrier within the Antioquia kindred. Genetic sequencing revealed this individual possessed an exceedingly rare missense mutation in a completely different gene, known as the RELN-COLBOS, or H3447R variant. This patient completely evaded the expected timeline. Instead of experiencing cognitive decline in their 40s, they survived cognitively intact well into their 70s. This side-by-side comparison of two brain PET maps reveals the biological paradox. On the left is a standard symptomatic PISA carrier, showing expected high levels of both amyloid and tau pathology. On the right is the COLBOS patient, exhibiting an amyloid burden 48% higher than typical symptomatic carriers. Despite carrying nearly double the amyloid load, the downstream pathology in the COLBOS patient is completely absent. Their entorhinal and global neocortical tau tangle burden was nearly zero, matching levels typically seen in healthy young adults. This case demonstrates that runaway amyloidosis can be mechanically uncoupled from cognitive decline. Massive plaque accumulation does not inherently guarantee neurofibrillary tangles or network collapse. Determining exactly how this single amino acid substitution insulated the brain against extreme amyloid stress provides a specific molecular target for uncoupling amyloid pathology from cognitive decline. To understand the rescue mechanism, we have to look at what normally protects the adult synapse. The reelin signaling pathway is the continuous, dynamic regulator of synaptic plasticity and dendritic spine formation in the mature mammalian brain. Reelin is a massive 450 kilodalton extracellular matrix glycoprotein. Its functional capacity relies on two distinct structural regions, an N-terminal dimerization domain called NR2, and a central receptor binding sequence spanning repeats R3 through R6. This structural model of the tripartite synapse demonstrates the strict requirement for oligomerization. Full-length reelin molecules must bind together at their NR2 domains to form large multimeric complexes. This massive structure is required to successfully pull together or cross-link the ApoE-R2 and VLDLR receptors on the postsynaptic neuronal membrane. This receptor interaction requires an obligatory electrostatic scaffold. Cell surface heparin sulfate proteoglycans, primarily the syndecan family, act as a multivalent trap embedded in the extracellular matrix. These syndecans possess long, highly negative N-sulfated heparin sulfate chains. These negative charges forcefully grip the basic regions of the reelin glycoprotein, locking it into the exact topological configuration required for receptor clustering. Once the extracellular scaffold successfully clusters the receptors, an intracellular cascade initiates. An adapter protein called disabled 1, or DAB1, is recruited and rapidly phosphorylated, which in turn triggers the activation of the PI3K AKT kinase pathway. Active AKT provides the critical neuroprotective payoff. It applies a continuous inhibitory phosphorylation to the enzyme GSK3 beta. By keeping GSK3 beta suppressed, the neuron actively prevents tau from hyperphosphorylating, stabilizing its microtubules and entirely halting the formation of neurofibrillary tangles. The collapse of this protective pathway begins long before plaques form in the cortex. We must trace the pathology deep into the brainstem, to the locus coeruleus, where noradrenergic neurons undergo early, severe apoptosis, severing their widespread axonal projections. The locus coeruleus is the central nervous system's primary source of norepinephrine, projecting massive arborizations that regulate arousal, stress modulation, and high-level cognition. The widespread degeneration of these specific neurons and the resulting systemic loss of norepinephrine tone is among the earliest neuropathological events in Alzheimer's disease, frequently preceding significant amyloid deposition by decades. Norepinephrine functions as a critical systemic immunomodulator. In a healthy brain, it binds continuously to beta-2 adrenergic receptors on resident microglia, actively suppressing the transcription of pro-inflammatory genes. When locus coeruleus neurons die, this endogenous immunomodulatory restraint is completely lifted. The chemical brake holding the brain's immune system in check disappears. Deprived of norepinephrine, the microglia undergo a rapid transcriptomic transition. They immediately shift from a resting, homeostatic surveillance state into a chronic, highly vigilant, and amoeboid reactive state. This technical flowchart illustrates the biochemical output of that shift. Once the unligand beta-2 adrenergic receptor prompts the microglial transition to the reactive state, the cells massively upregulate the synthesis and secretion of extracellular proteases, primarily matrix metalloprotease 9, or MMP9. Locus coeruleus degeneration initiates a brain-wide shift in the neural microenvironment, unleashing a localized proteolytic storm that begins aggressively digesting the structural foundations of the neural extracellular matrix. The flooding of the extracellular space with MMP9 initiates a targeted enzymatic attack on the rigid matrix structures that stabilize interneurons and projection neurons alike. This excess MMP9 directly attacks the reelin glycoprotein where it sits within the synaptic space. This animation maps the cleavage event. MMP9 acts as a highly efficient sheddase, slicing the 450 kilodalton reelin protein precisely between the R2 and R3 repeats, severing the required NR2 dimerization domain from the central binding unit. Without the NR2 domain, reelin is physically incapable of dimerizing. The resulting monomeric fragments bind to the receptors but act as competitive antagonists. They occupy the binding sites while entirely failing to pull the receptors into functional clusters. Concurrently, a secondary proteolytic attack occurs at the membrane. MMP9 acts on the syndecan family of heparan sulfate proteoglycans, shedding their core proteins entirely off the neuronal surface. This shedding is exponentially accelerated by endoglycosidases, such as heparanase, which are also upregulated by neuroinflammation and actively trim the protective heparan sulfate chains, making the syndecan core highly vulnerable to MMP9. The loss of these cell surface syndecans results in a total systems failure at the synapse. It completely removes the indispensable electrostatic scaffold required to concentrate and trap reelin. The result is fatal to memory consolidation. With the syndecan scaffold destroyed and the reelin ligand fragmented, intracellular DAB1 signaling goes silent. The inhibition on GSK3 beta is released, directly driving rapid tau hyperphosphorylation and the relentless spread of neurofibrillary tangles. Understanding this proteolytic destruction explains exactly how the RELN-COLBOS patient survived it. The precise biophysical mechanics of their H3447R variant allowed their synapses to operate inside this aggressive environment. This diagram of the reelin C terminus isolates the single nucleotide polymorphism. A mildly charged histidine residue is substituted with a strongly basic arginine at position 3447. The introduction of this basic arginine vastly enhances the local positive charge density across the reelin C terminus. Surface plasmon resonance data quantifies the physical impact. This charge creates supernormal electrostatic complementarity with the negatively charged heparan sulfate groups. It drops the dissociation constant from a wild-type 17 nanomolar down to an ultra-tight 10 nanomolar. Rigorous biochemical assays confirm this gain of function does not alter how reelin binds to the ApoE2 or VLDLR receptors. The enhanced affinity operates exclusively through the heparan sulfate co-receptor pathway. This tighter binding provides the mechanistic rescue. The enhanced electrostatic draw traps the reelin oligomers onto the remaining syndecan scaffold with extreme velocity and structural security. The mutant reelin essentially outpaces the disease. The supernormal affinity establishes and locks the signaling complex together faster than the upregulated MMP9 and heparanase enzymes can degrade it. By physically outcompeting the proteolytic storm, the COLBOS mutation forcefully sustains intracellular DAB1 phosphorylation. This keeps GSK3 beta firmly suppressed, mechanically protecting the brain from tau pathology even as amyloid plaques blanket the cortex. The kinetic data extracted from the COLBOS variant requires a necessary reevaluation of the historical amyloid cascade hypothesis. While amyloid beta accumulation exerts distinct toxicity, the COLBOS patient proves it is biologically insufficient to drive clinical dementia independent of extracellular matrix degradation. In this paradigm, cognitive decline is driven not by the mere presence of plaques, but by the cascading failure of biochemical pathways maintaining synaptic stability. This exposes a critical flaw in historical drug development. Clinical strategies aimed solely at clearing amyloid plaques entirely neglect the downstream structural and biochemical collapse already initiated by microglioproteases like MMP9. Efficacious treatment requires a new therapeutic mandate based on the extracellular matrix paradigm. First, pharmacological treatments must intervene directly at the membrane to forcefully stabilize the reelin heparin sulfate coreceptor scaffold, ensuring the synapse retains its capacity for plasticity and memory consolidation. Second, interventions must address the initial noradrenergic withdrawal from the locus coeruleus to prevent reactive microglia from releasing extracellular proteases in the first place. The RELN-COLBOS variant stands as biological proof that stabilizing the reelin signaling axis is sufficient to stop the clinical progression of the disease in a patient genetically programmed to lose their memory by age 50. Moving beyond mere amyloid clearance to actively target and protect the structural integrity of the reelin signaling axis offers a viable, mechanically proven path to finally arresting Alzheimer's disease.